All right, you want to start, Laura?
Yeah, sure. Hello, everybody. So we're going to start the seminar with a few announcements. As you know, financial assistance is available from the department for eligible students enrolled only in the thesis programs. To continue receiving this support, students are required to present their project at least once at the seminar. We have some available dates that you can book to present your project. It's a very good exercise, especially for the students in close for the graduation. So these are the winning students presentation for the last year. As you see, there are some awards involved. We also had a seminar question award, and we expect that award was offered for the students who put at least one question per seminar. So please be interactive and try to answer questions and find more information from our lecturers. We are also actively seeking out presenter and topics for the seminar. So if you have a topic in mind or you have a presenter that you think it will be beneficial for for the audience and for your project, please submit your idea to my email address, and we'll try to invite the speaker. So I think these are my announcements for today. We can invite Dr. Paxton Mwale to present our lecture.
Yeah, so thank you very much, Laura. Good afternoon, everyone. It is my distinct pleasure to introduce today's speaker, Dr. John Sampalis. Dr. Sampalis is a tenured professor of surgery and medicine at both McGill University and the University of Montreal. He's a globally recognized leader in clinical epidemiology with extensive expertise spanning clinical trial design, health services research, health economics, and outcome assessment, as well as biostatistics. Throughout his distinguished career, Dr. Sampalis has received numerous scholarships from major funding bodies such as the Medical Research Council of Canada, which is now called CIHR, the National Health Development Program, and the Fonds de la Recherche en Santé du Québec. His contribution to the field are reflected in his impressive track record. He has over 200 peer-reviewed publications in high-impact journals and more than $15 million in research funding. Dr. Sampalis has also provided invaluable support to numerous biotech and pharmaceutical companies, assisting in product development from phase 1 to phase 3 clinical trials and contributing to post-marketing strategy. He is particularly known for his innovative approaches to clinical trial design, including the implementation of adaptive designs and the use of synthetic controls. His research expertise spans multiple therapeutic areas, including oncology, immunology, rheumatic diseases, surgery, diabetes, and dermatology. In addition to his work in clinical trials, Dr. Sampalis has been a pioneer in real-world evidence, significantly advancing both its methodology and its practical applications to change healthcare practices and policies. Currently, Dr. Sampalis' work continues to align with the evolving landscape of evidence-based medicine, focusing on the integration of real-world evidence with traditional clinical trials, a crucial step forward in advancing patient care. Dr. Sampalis has also served as a consultant to various pharmaceutical companies, hospitals, and government organizations, and is a fellow of both the American College of Epidemiology and the American Heart Association. Please join me in welcoming Dr. Sampalis as he presents on today's topic. Dr. Sampalis, please.
Thank you very much, Paxson. Indeed, very kind words. Thanks to everybody for coming to the seminar today, where we are going to discuss and present some advanced statistical issues and challenges we might be facing in our everyday research practices. So I'll start sharing my screen. Um, is it visible to everybody?
Yes.
Okay, let me just this. Okay,
Maybe you can put in presenter mode, John.
yeah, I could try that. Let me try and see. Oh, see now I need to switch.
Yeah.
Um,
Yeah.
is that good?
Yeah, that's good.
Okay, fantastic. So what we will try to cover today are the following topics: missing data and what to do when we have them in our studies, if anything, survival analysis, time and event analysis, and prognostic covariates changing over time, analyzing the results before the studies end, or if we have more than one important outcome that we need to look at, and what impact do these multiple analysis could have on the sample size that is required for our clinical trials. Starting with missing data, this is a problematic issue in clinical trials and observational studies. The first question that we need to ask ourselves is what variables have missing data? If it's the treatment, it should never be in a randomized clinical trial. You could never have a situation in a randomized clinical trial where you don't know what treatment the patient received. Exposure to treatment as a missing exposure to treatment is also very rare in observational studies. But when it does occur, we should never replace. We should, in fact, ignore that observation and exclude them from the analysis. However, with respect to outcome variables or covariates, and especially outcome variables, uh, missing data missing data are an issue in both randomized clinical trials and observational studies. When we have this occurrence, this phenomenon, we need to make decisions on how to handle them. One question that should be asked, but is often never asked, is what is the nature of missingness of the results? And the nature of the missingness of the results could affect what we are doing or what can we do uh, in replacing the missing data. As an example, if patients miss their follow-up visits or if they are not doing well or because of an adverse event, then this occurrence of missing information in these follow-up visits is not a random is not random and is in fact impacted by the lack of efficacy of the treatment that their patients that are receiving or because of safety issues. We need to be very careful in situations like this to avoid the healthy subject effect by which only the subjects that are doing really well continue to be in the study and don't miss their outcome assessments. The nature of missingness. We have three situations or three classifications of how the data could be missing in a randomized clinical trial or an observational study. The first one is missing completely at random. This means that there is no predictive model that we could apply or no association that we can find between missing data and what we have as observed cases in the studies. In this case, we can conclude that the complete cases are representative of all cases randomized, and therefore the missing information may not have a significant effect on the validity of our results. Missing completely at random is very difficult to prove and validate unless you have very large data sets that would confirm this conclusion. The most common occurrence or the most common type of missing data is what we call missing at random. This means that the recorded or observed characteristics of the variables can account for differences in the distribution of missing variables in the complete and missing cases. What this means is that you could use the information that you have in the observed cases to determine the nature of the missing data or to explain why these data are missing. As in the example that I mentioned earlier, if the data are missing because of lack of efficacy, you could detect this and take it into account when you are trying to replace the missing data. The worst case scenario is missing not at random, which means there is a very strong association, something that's causing these observations to be missed, and this presents a serious problem in replacing them. When we look at missing completely at random, what we can do is we can say that the outcomes for subjects with missing data or those that dropped out are the same as for those that had no missing data or did not drop out. When we have missing at random, we can conclude that the outcomes for subjects with missing data or those that dropped out are the same as for those that had no missing data or did not drop out that have the same baseline or interim characteristics. Therefore, in this case, missing values can be estimated from those that we have observed. So you can say if you build the profile of all the missing data and find similar profiles of patients in your study with non-missing data, you can use the information from the non-missing data to replace your missing data. Missing not at random is, again, as I mentioned earlier, a bigger problem, in which case the outcomes for subjects that dropped out or are missing are different completely from those that did not drop out, even if they have the same baseline or interim characteristics. In this case, we cannot make any inferences of how to replace these missing data, these missing observations. So what do we do? Your obvious first choice is to do nothing, to do a complete case analysis. Analyze only what you have. This is not recommended as your only step because you have to make sure that there are no biases occurring when you are doing this. In other words, by ignoring the missing information, that you're not producing results that are not valid. In order for you to employ this step, which means to analyze only the available cases, you have to have verification that your data are missing completely at random. Even in this case, however, this analysis of only the observed cases does not take advantage of available information in cases with missing data that can be used to improve the validity of the analysis. So you are now ignoring all the missing information and you're doing nothing to replace it, and that could be very valuable information that you're not including in your analysis. Now we have simple imputation methods. One thing that I hear very often is, why don't we use last observation carried forward, or even worse, baseline observation carried forward? These methods of imputing missing data are very popular because they're simple and easy to understand. However, these are not recommended unless certain conditions and assumptions are fulfilled. I'm showing here some examples of what this may look like. Here we have an example for a simple imputation method using last observation carried forward. As you can see, for this patient, we had observations at month 1, 2, 3, and 4, but no observation at month 5. If we use last observation carried forward, it would mean the 5th month observation would be equal to the 4th month observation. However, if we looked at what the truth may have been, as you can easily infer from the slope that we are seeing here, assuming that there's no change between month 4 and 5, will introduce a bias in your results, and this could affect the overall results of your study, especially if the proportion of missing information is substantial, around 5, 10, or even 15% sometimes. So unless you could be certain that there is no change between month 4 and month 5, or a very low likelihood of a significant change between these two observation points, then you should avoid using this method of imputation. The other simple imputation method that is often used is known as baseline carried forward. Here we have the same example. This is what would happen if you imputed the baseline information, which is month 1. As you can see from here, you have a significant bias by applying this method. So when can we use last observation carried forward or baseline observation carried forward? Unless you only have 1 month, 1 follow-up visit, I never recommend using baseline observation carried forward. Last observation carried forward, as I mentioned earlier, can be used only when we can demonstrate there's no significant change in the observations between the period that you had an observed value and the missing point. In order to do this, you probably have to analyze the data from all your observed cases to see if there were any significant changes between those 2 time points. If not, then I recommend not using this approach. Now we have other methods that are being used. Single value imputation methods, where you use only 1 value to replace the missing data. This is a little bit better than last observation carried forward because it uses information from the observed cases and you replace, let's say, the missing value with either the mean or the median for continuous variables or the mode for the categorical variables. You can even stratify by group, or you can stratify by different other factors such as age, category, or gender, and replace appropriately with the mean or the median. This isn't as bad, and if you have no other choice, this is probably preferred to something like the last observation carried forward. So these are now the single value imputation methods that we just reviewed. In this case, we're using the mean replacement. Here we have a situation where we have 3 treatment groups, standard of care, new technique, and modified standard of care. We had 100 patients originally in the study, of which we had follow-up for 81, 81, and 85 with missing of 19, and 15 patients. The mean in these observed cases, as you can see here, is 8.22, 6.77, and 7.74. If we use the mean to replace all the missing data, we would see that the mean would stay the same. So very little bias here from the original, right? But if we looked at what the actual was, because when I did this, I randomly selected patients who have missing outcomes. And this is now the truth. So here you see what we imputed was 8.22. The actual was 8.19. We imputed 6.77, 6.52 as the actual, 7.74, and 7.73 as the actual. Not very bad. So we have minimal bias by using the mean in this situation, and this could be perfectly acceptable. What are the advantages of the single value imputation methods? They're easy to use, minimizes bias, and maintains the variance. You increase your power of the study because now you have more, a larger sample size, so therefore your power becomes better. However, what are the disadvantages? The disadvantages of the single value imputation is that they don't take into account the information from other variables that can be predictive of the missing values. And It usually requires missing completely at random and cannot be used for missing at random situations. Recall that missing at random is when there is an association between the missing values or the probability of having a missing value and some other variables in your data set. So when you can demonstrate missing completely at random, it's perfectly acceptable to use the mean, the median, or the mode to replace your missing data. Now we go to something a little bit more advanced. Here we use single value regression imputation methods. What do we mean by this? Well, it's based on this missing at random concept where we develop models to predict the missing values as a function of other variables. We use the observed cases to develop a predictive model equation for the target variable, and then we use the model equation. We plug these values into the cases with missing values, and we predict a missing value using the predicted value. Here I'm showing you an example. Okay, here we have equations that allow us to predict the missing variable, which is in this case length of stay. For the standard of care group, we have this model. This is a linear regression model. For the new treatment, we have this linear regression model. And for the modified standard of care, we have this regression model. And in this model, we have female coded as 1 and male coded as 0. So if you take every patient and you apply this predictive model to that patient that has a missing value, you can predict what their length of stay would have been. So if we go back here, you would apply these models to these 19, 19, and 15 patients to come up with their length of stay estimated or imputed values. This is what happens when we use now our regression model. Here we have 8.21 compared to 8.19, which was the actual, 6.76 compared to 6.52, and 7.74 compared to 7.73. Not too bad, actually pretty good. In fact, in this particular example, we can see even that when we use the mean to replace the missing values, our bias wasn't so bad. This is probably because the data are missing completely at random, or much of them are missing completely at random. Okay. Now we go to something a bit more fancy, which is known the multiple imputation method. And whenever I mention this to people, they go into a panic attack because it sounds like it's really very complicated, but it's not. Instead of using a single value to predict or to impute your missing variable, multiple imputation says you should accept the fact that for that missing observation, that missing value, there's some variability around it. That you can't really assume that it's just one number because you don't know what that number is. And that number is selected from as a random selected value from an infinite possible population of values of which you don't know what they look like. So in order to avoid this, we can say, okay, let's use a range of values. The most common way to do this is to run the regression models that I showed you earlier, or the replacement using 95% confidence intervals for the mean as an example. So instead of replacing it only with the mean, you replace also with a 95% lower bound and a 95% higher bound, and you run your analysis using these extremes, and then you come up with an average of that value to end up with your imputed value. You can use different regression models. In the example before, I used only a linear regression model. You can use a cubic, quadratic, or you can have the software figure out the best regression model to use for your prediction. You can use different sets of predictors. In the example I gave here, I used age, gender, and BMI. You can start with age, then you can add age and gender. You can add age, gender, and BMI. You can add any other number of variables until these models become too saturated to yield any meaningful results. And that's all of these things, by the way, they're slightly complicated statistical exercises, but what I intend to present to you today is the concept that is behind them, not necessarily suggesting that we will all be able to learn how to do these after this seminar. So here we see what happens with multiple imputations, right? I used a multiple linear regression imputation where I allowed the computer to run several linear regression models using randomly selected parameter estimates that are within the 95% confidence intervals of the parameter estimates that were predicted by each one of the linear regression models. And here we see what happens when we impute. We have 8.18, 8.19 as the actual, 6.59, 6.52, 7.73, and 7.73. We actually did very well with the multiple imputation method. So here's a summary of our imputation methods. You start with an incomplete data set. You replace missing data with a single value, and you run your analysis. The problem here is that single predicted estimated values used to replace the missing values do not take into consideration that there's variability around that missing value. Okay? Here's the multiple imputation method. You start with an incomplete data set. You run your imputations. Usually, I run about 5. If I have large data sets and a lot of missing data, I can go up to 15 or 20. It doesn't matter because the computers do these things now. Then you summarize, you pool all these data, and you run your analyses. So I'm just presenting to you a summary here about the different methods that we use to replace missing values. This is the actual value. This is the observed value when we just used only the observed cases. This is what we got with replacing the mean. This is what we got when we used the regression. This is with multiple imputation. In this example, all of these estimates are very close to each other. However, it's not uncommon to find that some of these methods yield completely different estimates. So what do? The first thing to determine is whether you have missing completely at random, missing at random, or missing not at random. You could do this visually. However, there are statistical tests that you can run to see whether your missing data falls into any one of these categories. Then you need to examine why the data are missing. Then you need to compare the missing and observed cases for other observed predictor variables. If you can conclude that your missing data are completely at random, you can use any method. If your missing data are at random, you prefer MI. And if your missing data are not at random, you have to use multiple imputations. You could conduct then sensitivity analysis comparing the different methods. Then I always suggest that we conduct what is called a tipping point analysis. You vary missing data from lowest to highest range in different proportions of patients to see if there is any effect on the direction or the statistical significance or the conclusion of your study. Why do we need to do this? And it surprises me how often this is not done. One of the questions that we are asking and one of the concerns that we have with respect to missing data is what could be the impact of the missingness on the conclusions and the results of the study. In other words, are we reaching these conclusions, let's say that the treatment works, only because the patients that were very sick dropped out and did really, really bad? And if those patients had been included in the study, then we would have concluded that the new treatment doesn't work, right? Tipping point analysis is a tedious process, but it's not very complicated. What you need to do is to ask yourself the question, what would happen if the missing data are biased in one direction or another? And what would happen to the conclusions of the study? What you do then is you replace the missing data with extremes of possible values and repeat analysis. Then then you ask yourself the question, or you determine how many or what proportion of the missing values need to change so that the conclusions are different, or the statistical significance is lost, or the direction changes. Let's take our example here. I'm just going to work with two groups. In the standard of care group and the new technique, we had 81 patients with observed length of stay. We had 19 missing in each one. As we saw before, the mean in the observed cases was 8.22 and 6.77 respectively, with a 95% confidence intervals showing here. If we run an independent student's t-test for the observed cases, we see that the difference is in fact statistically highly significant, with a mean difference of 1.45 days in favor of the new technique. Now, what would happen if I'm doing now a tipping point analysis in which I'm replacing all the missing data in the new technique with the upper 95% confidence interval of the standard of care? Why am I doing this? Because what I'm saying is this is the worst thing that could happen in the patients that received standard of care. So what would happen if everyone that dropped out in the study, or we don't have data on the study, did as bad as the worst case in the other group? So I'm taking this value and putting it in there. This is the results I'm getting. I'm still having missing data here, right? Now we see that the mean here, of course, stays the same, 8.22, but here I go from 6.77 to 7.14, a change, right? Here I have the comparisons, but here again I see that even with this replacement, the results remain statistically significant. The mean difference has changed, become less, but yet I can still conclude that the new technique is superior with respect to reduced length of stay compared to the standard of care. This is one-sided worst case scenario. The next would be a two-sided worst case scenario. In this case, I replace the missing data in the new technique with the worst case of the standard of care, and I'm replacing now the missing data in the standard of care, my comparison group, with the best situation that could have occurred in the new technique. Okay? 6.22 here and 8.72 for this group. Right? Now when I run the analysis, I see that this mean dropped from 8.22 to 7.84, and this one increased from 6.77 to 7.14, which is what we saw last time. I run now my t-test again. Of course, I see that the mean difference has been reduced. It's natural. However, what I'm observing here is that my statistical significance dropped, or increased, I should say, but it still remains statistically significant. What am I concluding from all of this? The results of my tipping point analysis have shown that the conclusions of the study will not be affected by missing values. This is what you need to show. Okay. Therefore, any imputation that I do simply improves the precision of the study and the validity of the estimates statistically speaking, but not from a conclusion perspective of the study. If the results, however, showed that the statistical significance was lost or the direction of the difference was reversed, what I need to do then is I start changing a proportion of the patients of the missing data with those values to see where do I get a tipping point. And then at the point that I find where I get the tipping, I need to go back to the data and see what's the probability of these values being observed in each one of the groups. And you assess this conditional probability. Again, these are slightly complicated statistical exercises, and you need someone that understands how to do this, but it's important for all of us to understand what's behind these processes and make sure we pursue these kinds of assessments when we have missing data. Okay, we now get to the second part of the talk, which is on survival analysis. You're running a randomized trial testing the effect of treatment on COVID-19 on mortality. In 60 days, you enroll and follow the patients for 60 days. The outcome is all-cause mortalities. However, some patients drop out before the 60 days. How do we handle the dropouts? This is what we know in the sensor data. We do not know the survival status after the time of dropout. And within the 60 days that we're looking at, the window of observation, right? Some of them may have continued to live, but some of them may have deceased. Therefore, this censoring could cause a potential bias. This is what the data might look like. Here we have patients that are alive, but only followed for 29 days. Here we have a few more patients alive, 59 days, 49 days, and 49 days. Okay, these 59 aren't so bad. This 29 is a serious concern, and these two are also somewhat of a concern. Someone might say, well, why don't you just compare the proportions deceased? And yes, we can do that, right? Ignoring the fact that some of them weren't followed forever. And here we can see that there is a difference between the groups, 23% and 45%, however, not achieving statistical significance. The other thing that we can do is we can say, well, why don't you compare the time to death in these patients or the duration that they were alive? Here we see in the placebo 36 days, and here we see in the new treatment 53 days. Again, here achieving statistical significance between the differences in the mean. However, all of this analysis is biased because it's not taking into account the censored data. Both the chi-square and the t-test does not consider the fact that the incidence of death and the duration of follow-up could have changed during the period after the patients dropped out from the study. We need to estimate what could happen to these patients after dropout. We need an analysis that allows us to combine a continuous outcome, time to death, and a binary outcome, survival, which is yes or no. A solution to this is a survival function. This awful looking thing here with a lot of Greek in it is what is defined as what the equation that we use to calculate the survival function. Survival function, or the Kaplan-Meier estimate of the survival function, is simply a product moment estimate. And the way that you do this is you simply look at the point in time that the first death occurs. You figure out how many people were alive at that point in time. You calculate the probability of death. You calculate the probability of survival, which is 1 minus that. And here you have the cumulative probability. You go to the next time an event occurs, you repeat this, and then you multiply this by this to give you the cumulative probability. At the end of this, you're going to get the cumulative probability of survival at the end of your follow-up period. Everyone has seen these Kaplan-Meier curves. This is the cumulative probability of survival that we see. And this is the time of period of follow-up. In this example, we have 400 I think this should be, 400 months of follow-up. What is important when we're looking at these Kaplan-Meier curves is to look at, when I look at this, I look at two things. I look at the slope. What is very interesting here is that we have a really fast slope up until 50 months, and then we have a much slower slope. But this could mean that in this case, disease occurs really, really quickly in the first 50 months, and then those that survive live for a much longer time, typically almost 300 months that you can see here. So when I'm analyzing data like this, I'm very careful when I'm running some conclusions about treatments that I don't allow this component to affect my results, right? Because this is a survivor effect. When you're comparing two groups, you would like to see differences in this part. The important information that you're drawing from a single curve like this is what we call the median survival time. This is the point in time where 50% of the events have occurred. And in this case, you can see that the median survival time occurs at 50 months. If we take the same kind of analysis and we're comparing two groups, right? Referring back to what I talked before, here you can see that both of these groups have a rapid decline in the beginning, but then they seem to be tapering off. And this difference here could be really a difference because of the treatments. In this case, it's a difference between smokers and non-smokers. Here you could just draw a line here to see where the 50% marks are to compare the median survival times for smokers and non-smokers. And officially, how we do this comparison of the survival curves is something we call the log rank test, which is the comparison of the observed and expected survival functions, and you get a p-value using a chi-square to compare these observed and expected survival functions. And here we can see that there's a statistically significant difference between the survival time or the survival rates of smokers compared to non-smokers. Again, it's very important that you look at these graphs very carefully. In this case, we have nice parallel graphs here and parallel graphs afterwards, which gives me little concern that I have bias if I'm including the entire function. However, if I only saw in one group decline like this and then in the other group a more gradual decline, I would just focus on the period of time where I have a rapid decline, and then I would analyze the data separately for the time period after the rapid decline. These are some more examples of survival curves and survival analysis. In this case, I think this came from the COVID study. And here what we see when you do run this analysis, in this particular example, you will see that the medians can't be calculated. And the reason that the medians can't be calculated is because no one reaches the 50% point, right? Again, another thing to look at. In the previous example, we see that we reach the 50% point. In this example, you see that you don't reach the 50% point. When you don't reach the 50% point, you don't look at the median, but you look at the mean. And this is what we have in this example. The curves are still compared to using the log rank test. And again, here we see non-significance. Now, let me see if I have this. If you look, what I wanted to point out to you, if you look at these estimates for mean survival between new treatment and placebo, here we have 55.8 and 41.3. If we go back to the crude estimates that we produced here, you see that they are 38 and 53. They are completely different. The reason that these estimates here are different is because they take censoring into account, and these estimates are produced based on the cumulative probability of survival that is calculated using the Kaplan-Meier estimator. Why am I pointing this out? I'm pointing this out to tell you that you should never use students' t-tests or mean comparisons when you're looking at time to event analysis. It is completely wrong. Okay. Usually when we talk about survival analysis, we talk about this other thing, the hazard function. The best way for you to understand the hazard function is conceptually at a very high level that it's the complement of the survival function. Whereas the survival function looks at how many individuals don't have the event, and in the case of mortality, how many individuals are still alive. The hazard function looks at how many individuals have the event. A little bit more deeper into the definition of the hazard function, it gives us the probability of an event occurring at any point in time, t+1, given that the event that the individual is event free. There's a free missing here at the time t. So, in other words, if an individual is alive today, what is the probability that this individual will die tomorrow? This is known as the force of the hazard or the hazard function. And it's becoming very important because it has a log linear association with time that allows us to put it into models like Cox regression models. Why is this useful? It's because when we use Cox regression models, this is, by the way, the function of the hazard function formula. When we use Cox regression models, and in fact, Cox proportional hazard models, we can do adjustment for covariance. When you're looking at the Kaplan-Meier curves here, you can't do any adjustment. So even if your groups are different at baseline, you can't do anything with the Kaplan-Meier analysis. However, the Cox regression curves, you could. You can add as many covariates as your model will tolerate. Okay, so this is why it's beneficial in these cases to look at your Kaplan-Meier curves as descriptive, but to run your Cox regression models as more informative and giving you the opportunity to add covariates to your model. Let's take an example here. We're conducting a randomized clinical trial to compare two chemotherapies, old, which is the standard of care, and new regimens for the treatment of prostate cancer. You put 300 patients in the study and you try to enroll approximately all of them 1-to-1 ratio to the new and the old regimens. The primary outcome of interest is remission. You use survival analysis to assess time to remission between the two treatment groups. This is your Kaplan-Meier analysis, and you can see here, now we have estimates for the median because we get to the 0.5% mark, that 29% time to remission in the old group, 29 days time to remission in the old group, 23 days in the new group, and this is statistically significant, right? So you can expedite, whether this is, maybe this is months, you can expedite remission by, let's say, 6 months or 5.5 months when you're using the new technique. And when we look at the mean, we see again about the same thing, about six units. If this is 6 months or 6 days, I think this is 6 months. Important thing here is that this is now statistically significant, and you can see from the curves here that we have nice differences. We are not concerned about early and late effects here because we can see both treatments have the same kind of rate or slopes in the beginning, and they seem to be a bit more faster in the later part of the study, right? And this doesn't cause us concerns or any potential bias related to time. However, yeah, and this is just making this graph look a little bit better for you to see. Here we have the hazard function for these two treatment groups, and you can see, as I mentioned earlier, the hazard function is looking exactly the opposite direction of the survival function. Again, differences, and now Cox regression analysis shows us that there's a statistically significant difference between the two groups with respect to the hazard rate, the hazard ratio being 0.5 with 95% confidence intervals between 0.36 and 0.69, meaning that the patients in the new treatment have remission at a 50% or twice as much rate as the old treatment. Now, while you're doing this study, you have discovered that during the course of the study, immunotherapy was offered to all the patients. And you're asking yourself, what is the effect of the duration of immunotherapy? What is the effect of immunotherapy on the results that I observed? What effect could this have on the differences between the two treatment groups that I analyzed? The problem here is that immunotherapy was not started at the same time for all patients. Now, this becomes problematic because you can't classify any particular given point in time, let's say the first 3 months, in every patient to be without immunotherapy and then immunotherapy, because this varies from patient to patient. This is not a covariate that varies with time, or is known as a time or time-dependent covariate. And the time on immunotherapy will be different for each patient. You now need Cox regression with time-dependent covariates. What is the solution? You need to create a variable that defines the time to immunotherapy for each patient. Then you need to run Cox regression with time-dependent covariates in which you will assess the remission rates before immunotherapy and after immunotherapy. This is what these results look like. As you remember, we have remission in the old and new. We have 41% remission in the old and 61% in the new treatment. Time to immunotherapy was about the same for both groups, so we're not concerned that one group got immunotherapy faster than the other one. In the first part of the analysis, you will define your time-dependent covariate as a time before the start of immunotherapy. You put this into your model. Then you run Cox regression with time-dependent covariates with group, and group times, the group is the main effects variable here, and group times the time covariate interaction in the model. The conclusion here is that the hazard rate before the time of immunotherapy, before starting immunotherapy. So if we were to focus only on the time before immunotherapy was started in these patients, is 0.669 with a p-value of 0.07, which approaches statistical significance. This is not negligible. You still have a 30%, 35% better remission rate in the new treatment group compared to the old treatment group. So this isn't bad. Now, you have to do the opposite. Now you have to define now you have to define the time-dependent covariate as the time after you started immunotherapy. And you run the analysis again here. What do you see here is that now the hazard ratio becomes 0.376, which is highly statistically significant. The results of this analysis show that the remission rates improved with treatment after starting immunotherapy. And in order to come to this conclusion, we used a time-dependent covariate to assess the effect of immunotherapy that started at different calendar points or different points in the study for each patient, and the duration of immunotherapy, or the duration of the time since the patient started immunotherapy on the results. Based on this analysis, you will conclude that while your new treatment works, it seems to work a little bit better when you add immunotherapy to it. Of course, this could be an expected result clinically, but it's interesting when we're able to prove it with this analysis that takes into account the time-dependent covariate. Now we're going to go to the third part of the presentation, that talks about what we do when we have multiple analyses or multiple outcomes. I just want to refresh everybody about what happens in the research process. We start with a target population from where we get a study sample. We run our statistical hypothesis on the study sample. We conclude on the study results based on the study sample. Then we try to make inference back to the target population. Because we are using a sample here, we introduce sampling error because we may not be getting the best representative sample of the target population, especially if the sample is small. Your sampling might be biased. So you're introducing sampling error. You run all your statistical analyses to assess how much sampling error can you get. And then you come here to make your inferences. During the inference process, you have to correct your sampling error or quantify it. Say I have a probability of this much of making a sampling error. Sampling errors are the results of hypothesis testing errors, or sorry, sampling errors cause hypothesis testing errors. When we talk about statistical hypothesis testing, we are all familiar with the fact that we either reject or fail to reject a null hypothesis. When we reject a true null hypothesis, we commit a type 1 error, and when we fail to reject a false null hypothesis, we commit a type 2 error. Put in another way, what you really want to do is not commit an error, but when you commit these errors, this is what they're defined as. When we talk about alpha levels, it's the probability that we are committing a type 1 error. And we're talking about beta levels, we're talking about the probability that we are committing a type 2 error. The power of the study is defined as 1 minus beta, which is the complement of the probability of committing a type 2 error. Essentially, the power of the study is letting us know if we are able to detect a true association when it is there. Conventionally speaking, we pick type 1 error as 5%. This is this famous 0.05 that everyone is chasing after when they're doing research. And most people, when they pick up a journal and they read a clinical trial, they go to the table and they look if the p is less than 0.05. And unfortunately, some people, even in regulatory agencies, can't see beyond this 0.05. What I always emphasize to everyone is that this 5% is just a probability of a chance that we conclude that the treatment works, as an example, when in fact it does not work. It's an arbitrary number, and it could be affected by a whole number of things, including how we sample the results, what analysis we do, what variability we have in the results, etc. And it's not the absolute gold standard definition of whether something works or not. It's just a statistical indicator. And as a statistical indicator, it should be only confirming or not confirming what we see as clinical importance in our studies. If the results are clinically important and yet statistically not significant, you need to examine whether you have enough power, whether there's something wrong going on in your study that prevents you from getting statistical significance. Let's talk now about what happens when we split this p-value. As I mentioned earlier, the p-value is the probability of you committing a type 1 error or having a false positive result. Imagine that we have this situation where in 100 different repetitions of the experiment, we have a 5% chance of finding a study or doing a study with a false positive. So if we only do one study or we only do one analysis, the probability of a false positive is 5%. But what if we do 2 studies or draw 2 circles? Then it becomes 10%. What if we draw 3 circles? It becomes 15%. Every time we run a study, every time we pick a circle from this population, we increase the probability of committing a type 1 error. But by the baseline probability that exists in the entire population. To put this as an example, you have decided to execute the previous study on new chemotherapy, and you have been told that you're going to enroll 40 patients per group that you will treat with your new drug and SOC. You will assess 2 years progression-free survival in this case. And the patients that are withdrawn or expire before the 2 years will be considered as not having achieved 2-year progression-free survival, and therefore you don't need to recruit more patients to compensate for early withdrawal. You have accepted that a 5% chance for your study of false positive is what is acceptable to you, so you've determined your alpha to be 0.05. And you're saying that you're taking a 5% chance that you will conclude that the treatment works better than SOC when it really does not. This is your limit for false positives. That is it. That's your speed limit. That's your limit. You can't go over above that since you now have a priority defined it. However, you realize that it will take 2 years to recruit the 80 patients, and you need to follow them for 3 years, so you won't, 2 to 3 years, so you won't be able to know the study of the results for 4 or 5 years after accounting for startup, etc. This is too long. So now you say, okay, I'm going to run the analysis, when I have 10, 20, 30, and 40 patients in the study. So interim analysis when I have these many patients per group. And you say, well, if I find a statistically significant effect in any one of them, I can conclude that my treatment works better than standard of care with a 5% false positive chance. But this is not true because you have exceeded your false positive limit because you've done this 4 times, so you are now actually at a 20% total alpha. You're only allowed 5%. What do? Find a biostatistician and ask them what happened because they told you you can enroll 40 patients in the study. The problem is that you have a maximum of 5% false positive probability, but you will do more than one test. Now, here are the solutions. The easy solution is the Bonferroni one. I call it the Italian solution, by which you divide your total allowed false positive rate by the number of tests. In this case, we had 5%. You divide it by 4, 1.25%. So you say, if for any one of my analysis, I find a p-value less than 0.125, 0.0125 or 1.25%, I will conclude then that the new treatment works better than standard of care. This is easy. However, the p-values that we end up trying to achieve here are very small. And sometimes if you want to do 4 or 5 analysis, that p-value becomes 1% really quick. And in order to achieve 1%, it's very hard, or you have to enroll a lot more patients in the study to do this. And this is sometimes not possible. Now you need to be a little bit smart about this. We all know that as the sample sizes become smaller, the power becomes less, and the probability of seeing a statistically significant difference decreases. So the question then is, why will I spend all my p-value or one quarter of my p-value when I'm doing only when I'm only doing 10 patients, right? When I know that with 10 patients in group, I have a very low chance of achieving that p-value. So why do I spend it all there? Why don't I conserve some of it and spend a lot of it later on when I have much more power in my studies? And here you can see how your power increases with the sample size. So I spend it here when I have little power to detect it, and not save it and try to spend it here. But here at least I can take a peek. Well, the requirements are, remember that your total alpha does not exceed 5%. What we can do then because we can divide the total alpha in other ways that reflect the difficulty of getting statistical significance and preserve it for analysis with higher power later on in the study. We must take into account the following: that we want to terminate the study as early as possible if we can conclude that either drug works, or that we would like to stop the study early in case of futility, which would mean we conclude that no matter what happens, there is no way that we can demonstrate that the new treatment works better than standard of care. This becomes an ethical concern, and many times regulatory agencies require that you put a futility assessment in your study to make sure that you're not enrolling patients in the study for which there's no possibility or very little possibility that you will demonstrate that the new treatment works. The total alpha conventionally covers both sides, efficacy and futility, but efficacy is dictated by the power of the study and therefore the beta values. We have several options how to spend our alpha this way. The most commonly used way of breaking up the alpha at different points in the study is the O'Brien-Fleming approach. And this is what you would get when you would apply your O'Brien-Fleming approach in the example we talked about before. When you enroll your first 10 patients, your target p-value would be 0.007, which is hard to achieve, but at least it gives you a glimpse. When you bring in the other patients, it gets better. 30 patients, it gets better. And then at 40 patients, see 0.02, right? And keeping in mind that this is for efficacy only, so your limit here would be 0.025, not 0.05, because we're only interested in the right side of the curve where the treatment is better than the standard of care. You typically get a graph like this with this kind of analysis, and it's telling you at your first analysis, if your test statistic falls in this part, then you have proven that the test drug works better than standard of care. Or if it has fallen in this part, you have proven that the test drug is worse than standard of care. In both of these cases, you will stop your study. Now, this is what it looks like when you get the results for futility analysis, because now you want to determine, is there a chance ever that I can show whether or not these two treatments are different? Or is it futile to even keep running this study because it will never happen? And when you add futility to your analysis, here is the area, and you can only run futility analysis when you have enough sample size, so it starts occurring somewhere around here. So after your second study, after your second analysis. So if your test statistic falls in here, you can conclude that your drug works better than standard of care. If your test statistic falls in here, you can conclude that your drug is worse than standard of care. However, if your test statistic falls somewhere in here, you need to stop the study for futility, because given the way the data are right now, there's no way that you will be able to demonstrate a difference between the two groups. So it's futile to continue the study. What happens now if, in addition to progression-free survival or overall survival, we also wanted to assess progression-free survival? In fact, someone might argue that for certain diseases, progression-free survival is more important than overall survival, since it takes into account disease-free state. If we test both progression-free survival and overall survival, we are inflating the alpha value of the study because you're running the analysis twice. So we need solutions to allow the assessment of both of these outcomes without increasing our type 1 error probability. In this case, now we have two hypotheses. Hypothesis 1 is that there's two null hypotheses. Hypothesis 1, that there's no effect on PFS, and hypothesis 2, that there's no effect on overall survival. The solutions to this is, again, The most easy one is the Bonferroni, weighted Bonferroni procedure, by which you test both hypotheses at 1/2. Now you could have 3 hypotheses, and this could become 1/3 or 4, could become 1/4. You take your alpha, you divide it by the number of hypotheses, and you test each one at 0.025 in this case. And if any one of them is significant, then you conclude that your treatment works better than standard of care. However, this is not ideal, as I said before, because it spends a lot of the alpha, and it puts equal weight for both of these studies now, for both of these hypotheses. So you may show that it works for overall survival, but not progression-free survival, so this may not be as interesting. Now we have something called the fixed sequence. This kind of approach is known as the gated approach. It's called the gated approach because you only allow certain things to happen after something else has happened. So it's a conditional procedure. And it's called gated because you identify which hypothesis is by far more important to you. In this case, you could say clinically, if I'm not showing progression-free survival, overall survival doesn't matter, right? So I need to first demonstrate progression-free survival. What happens in this case, you come here, you test your first hypothesis at 0.05. If this works, then you go ahead and you test your second hypothesis at 0.05. Because you've already done the work here at 0.05, so you've used that up, and you're no longer testing this hypothesis at the same time as that one, you can transfer your alpha levels to the second hypothesis and test the second one at 0.05 and conclude if your drug works or not. If you weren't able to prove that your drug works, in other words, if you couldn't reject the first null hypothesis, you don't get to the second one, right? This is a hierarchical approach or gateway approach that is really convenient because it allows us to look at the important outcomes first and then focus on the secondary outcomes without necessarily being penalized if we put 5 or 6 secondary outcomes in our study. Then there's a deviation of this procedure by which you first test the intersection hypothesis, which is both of these hypotheses at 1/2 alpha and 1/2 alpha. So now you say, okay, I'm going to test both of them, but each one at 0.025. If either one of them is correct, then I can move the entire p-value to the second one, right? This allows you to retain your overall p-value of alpha, but give a chance to each one of these hypotheses to be proven wrong or to show that it works. So sample size is a function of effect size, variability, alpha level, and beta level. Multiplicity adjustments affect your alpha level. To retain power and alpha size, sample size with multiplicity adjustments will be higher than without. Usually we see an effect between 1 and 40% depending on the type of adjustments and what function is used to split the alpha levels. I've given you an example here. Running a randomized clinical trial, we assume 40% remission in the old and 60% remission in the new treatment. We've established the power of 80%, alpha is 5%. If we don't do any interim analyses, we require 95 patients per group for a total of 190. If we apply the Bonferroni, which is the worst procedure we can go after, we need 282 patients, 141 per group. If we use O'Brien-Fleming, which is the better one, we only need 222 patients. If we use the Pocock method, we end up with 129 patients or 258 total, which is somewhere in the middle. The Pocock method allows you to stop the study earlier because it puts a bit more weight on the early interim analysis compared to the O'Brien-Fleming. There are other ways that you can do this. You can manually enter how much of your p-value you want to spend at each point. And that concludes the seminar. Thank you very much.
Thank you very much, John, for that fascinating seminar. So there's any questions? There's a question by Mashid. Mashid, would you like to unmute and ask? You are muted. Can't hear you. You're breaking. Okay, I can read your says understand the value of data and our efforts in keeping as much as participants as we can. However, when we should decide to drop a case from our analysis, especially in survival analysis.
When do you drop a case from survival analysis?
Yes. Can you hear me now?
Yeah, I could. Could you say that again?
Thank you so much for the great presentation. I know about the outliers that when we know the odd actually variable, the odd amount of something, but I just wanted to know, is it, for example, in 4-year follow-up of breast cancer treatment, and if we just have some patients like censorship after 2 to 3 months, do we drop them or do we keep them?
That's a very good question. You bring up a very good point. One of the first things that you need to look at your survival analysis is censorship pattern, right? So that's where you decide, in fact, what kind of analysis you will use. But that's also where you're going to pick up if in certain treatment groups, the censorship patterns are too early, let's say, right? Now that creates a problem, right? And then you have to go in and understand what happened with these censored patients. Why did they drop out, etc. If you only have 1 or 2 that drops out really quickly, then you could remove them from the analysis. But if you see a pattern being developed within one particular group, then you need to assess why these patients dropped out and make a decision on a case-by-case basis.
Okay, thank you so much.
Okay.
There's another question by Joshua Temitov. Great presentation. What is the difference between regression and imputation in missing data?
Oh, regression is one method we use for imputation. So, do you mean regression analysis like when you do generalized linear models? If that's what you mean, generalized linear models don't impute missing data. They estimate a general value that includes the missing observations, but they don't assign a missing value to the observation. It's like drawing a line between different points, and some patients might have a point there, some patients may not. They draw the line, and then you use the line to estimate it, but it doesn't assign a particular value to that patient.
Okay. So, John, as you were speaking, I was wondering, in terms of survival analysis, how does survival analysis address the issue of competing risks, where you have multiple events that occur, and the occurrence of one of event precludes the other? Let's say you have death from different causes.
Yeah, we model competing risks in survival analysis using a similar approach with a time-dependent covariate.
Yeah.
So you would you would consider that, sorry, you would consider that as almost an event of starting immunotherapy, right?
Right.
And you would put that in the model.
Okay.
It's a bit more complicated than this because the actual event could be linked to the outcome that you're looking for. So a stroke could precede death, right?
Yeah.
So and you need to make sure that the timing sequence is appropriate. So, but it's similar to that.
Okay. Yeah, so the other question was, how can machine learning techniques be integrated with survival analysis to improve prediction accuracy or identify some complex? Because as you are talking about prediction accuracy, I was thinking, can't machine learning help?
Well, a lot of these imputation methods, so when you're applying multiple imputation, you're in a way getting the machine to do the random allocation of the values, right? So in a way, it's the machine that's doing this. Now, you can develop machine learning algorithms that will run these analyses on very large data sets, and it could improve the prediction that we're getting through things like multiple imputation, but it's still, in my opinion, at least the same underlying process, except now you're using the machine to select the random variables using some other algorithm rather than the statistical software, which uses statistical algorithms to replace the values.
Okay. Any other question?
Sure. Hi, Dr. Sampalis. Very nice talk. Very informative, actually.
Thank you.
I just had a question regarding well, two questions, but the first one is regarding the missing data. So if you had like a situation where you're dealing with results that were diurnal, like how is that the same rules apply? Like, does that become more difficult to kind of predict, let's say, a missing value within a diurnal curve?
Yeah, it becomes a lot more complicated in that situation. And that's when you have to start looking at the data. And that's where, as I mentioned earlier, the first thing you need to do is look at the pattern of missing data and see what you can identify then. And then you might have to split it up and deal with it in different ways. But it's not as straightforward as this.
The other one was on the p-value splitting, which is kind of interesting. How do you know what method to use? Like, if you're going to go with the Bonferroni way or the Brian Fleming, you randomly test them and see, or do you have to like pick one up? I'm assuming, and then you stick with it until the end.
Well, so I use the Bonferroni test for sample size calculations because I want to be super conservative to bring in the highest possible sample size that I might need. Then often it becomes a decision on why you are doing the interim analysis, right? So if you're just doing the interim analysis to get a glimpse of what is in the data, but not necessarily intending to stop the study early, I would use the O'Brien Fleming because it spares a lot of the alpha for the end, right? But now if you have the VC pushing you and saying finish it as early as you can because we want to go to market and get our money out, then you push the alpha level earlier. It's always a risk, right? Because and I tell everybody, you don't get to come back to the table if you fail in the first interim analysis, right? So
exactly, yeah.
you don't okay. Well, now I changed my mind, and you call the bet. The die is cast. It's done, right? So I caution everybody. Yeah, I get it. It sounds a lot more exciting, but if you fail, you're done.
Got it.
Yeah.
So thank you very much, John, for that wonderful presentation. And thank you for all the help you gave our team when we needed your help.
Absolutely. Anytime. You all have my email address. Don't be shy. Ask questions.
Yeah. And thank you, everyone, for attending the seminar. Thank you.
Wonderful. Thank you
Yeah,
very
thanks.
much. Thanks for the opportunity. It was
Yeah.
great. Thank you, Laura. Thank you, Faxon.
Yeah.
Thank you. Bye-bye.
Bye.
Bye.